Very good morning, everyone. This is the Cogan Award presentation. I'm very pleased to be able to start this session. A very good morning to everyone and welcome to ARVO again. I'm Tian Wong, the Clinical Epidemiological Research Trustees. And it's my pleasure to talk about the Cogan Award, and this year's recipient. We will have another person that will speak about him. So let me review the history of the Cogan Award. The award was established in commemoration of the outstanding scientific contributions of David G. Cogan and recognizes a young researcher who is 45 years of age or less at the time of nomination. The Cogan Award recipient will have made important and worthwhile contributions to research in ophthalmology or visual science that is directly related to disorders of the human eye or the visual system, and who exhibits, of course, substantial promise for future contributions to eye and vision research globally. The 2025 Cogan Award is presented to Dr. Alex Hewitt, PhD, MD, PhD. He's a research fellow at the Menzies Institute for Medical Research at the University of Tasmania and a principal investigator at the Centre for Eye Research, Australia, University of Melbourne, Australia. His major research interests lie in the ophthalmic application of stem cell and gene editing technology, of course, very important now in ophthalmology. So it's really my great pleasure to welcome this year's Cogan Award. I've known Alex for many years, more than 20 years now, and it's my pleasure to welcome Dr. Alex Hewitt. Please.
Thank you.
I'm now pleased to also introduce Dr. David Mackey, Professor of Ophthalmology at the University of Western Australia, also a longtime friend and collaborator, who will review, of course, Dr. Hewitt's achievements and introduce the lecture. David?
Thank you very much, and it's a great honour and pleasure to introduce Alex Hewitt for the Cogan Award. Given that we have to ask a family history of patients that we're treating, I would like to point out that we're in the world capital of genealogy, Salt Lake City. And here we have part of Alex's extended family tree on his mother's side. Here he is on page 8. And also his children and his partner Meg, Bonnie, Ted and Rue. And this is the importance in genetics. Alex actually attended his first ARVO 21 years ago when he was just starting work with us as a PhD student and has been to most ARVO meetings since. Alex started his PhD. You often notice that he's wearing a backpack at ARVO, more appropriately in the wildernesses of southwest Tasmania. And Alex and I went hiking in the first few months of his PhD, and we had such a terrible rainstorm we were tent-bound for a day, and that's where we wrote our first grant together for the twins' eye study. Now, Alex is with his other PhD supervisors, Jamie Craig and Catherine Burden. Alex spent a year with me in Tasmania and then went to Adelaide, and this is the only photo we know of in existence of Alex actually wearing a jacket. Alex has done a lot of field work with our team around the world, including Norfolk Island. We don't have penguins there, but they did get a tariff slapped on them, as well as the Guangzhou twin study with Mingwang He, and also field work in the north of Greece, where we were tracing family members with mycelium mutations, where we'd found other relatives in Australia. Although Alex is always easy to talk into going to somewhere interesting and quirky as a tourist, so after the Academy meeting in San Francisco, we drove to Mendocino at night so we could stop on the side of the road where Kerry Mullis stopped in 1983 to write down the formula for the PCR reaction. Also, when we went to the World Ophthalmology Congress in Barcelona. We went south of Alicante to the salt lakes of Sao Paulo, and this was where Francesco Moya isolated the H. mediterraneae, which contained the CRISPR sequence leading to that whole area of research. Alex graduated from his PhD. He then did his ophthalmology fellowship, and he's back in Hobart, Tasmania, as Professor of Ophthalmology there. And here he is with his partner Meg when he qualified as an ophthalmologist. Alex has been an inaugural member of the International Glaucoma Genetics Consortium, and we have monthly teleconferences, but Alex actually held one of the meetings in person in Hobart, hosting it here with the key members of the team. But I think the best example I give of Alex as an innovative thinker was he was the president of the Junior Doctors Society in Hobart, and they had their annual dinner at the new Mona Museum, which is an amazing modern art and old art museum. And unfortunately, there was a bit too much revelry, and one of the major artworks there got damaged, and Alex was summoned as the president of the society to the owner of the museum, David Walsh, to say how he was going to repair or pay for this artwork. So Alex innovatively said, "You have an Egyptian mummy in the museum. Could we organise for that to have a CT scan done at the hospital?" This was a great idea according to the museum owner, who was very keen about it, as was the radiology department at the hospital. Unfortunately, the head medical officer of the hospital could just see the newspaper headlines of patients wait 5,000 years to get CT scans done at the hospital. So that was scuppered, but then Alex managed to convince the private radiologists to do the CT scan. Now, Alex has over 400 publications. He has over 30,000 citations and has an H-index on Google Scholar of 88 at such a young age. We're going to see a lot more work from Alex, but I will introduce him with great pride to give this 2025 Cogan lecture. Alex Hewitt.
Thanks, Dave. Thank you. Thank you for that introduction, David. I think I only get called Alexander by my mother when I'm in trouble, so it was fairly humbling to see that on the first slide. I'm also a little bit embarrassed to say that I don't think I'd actually fit into that suit jacket anymore. In terms of disclosures, these are my conflicts of interest that relate to this work. Now, in terms of the genetic testing, I was extremely excited when David pitched to me the prospect of sequencing a gene, and this was only a year or two after the first draft of the human genome had been published. And there was family hearsay that Meggie, my partner, was actually related to Manna Legena. The prospect of and the real power of population genetics really only shone through to me following this work. And essentially, Meggie and I submitted our DNA samples to 23andMe. And from that, we sequenced our genomes. So Manna Legena was the leader of the Ben Lomond tribe, which is in the north-east region of Tasmania. And as I said, there was a family hearsay that Maggie's great-great-great-great-great-grandfather was Manna Legena. And it was only through, interestingly, after getting the results from 23andMe that we actually confirmed that Meggie was actually 1/64th Tasmanian Aboriginal. By contrast, my genetic ancestry is certainly not as interesting. So I think this really underpins the potential power of genetic testing. And I think from the first sort of outset, the prospect of screening people in the population for identifying people at risk of developing disease. And given that glaucoma has such a high heritability, the prospect of integrating genetics into treatment and patient care, I think will be transformative for this condition. So the question gets raised then, why has it taken so long for any major impact on our understanding of the genetics of glaucoma to make an impact in the clinic? And I think that in part comes from our complexity and size of the human genome. For me to wrap my head around the actual size of your 3.3 billion nucleotides in your genome, I think one of the best analogies would be that if you expand the size of each nucleotide in your genome to be the size of a dime or a 10-cent piece, you would have enough coins that if you stack them end on end, you could cover all of the bitumen or sealed surfaces in Tasmania. Tasmania is the island at the south of Australia, and that's about the same size as West Virginia or Ireland. And then, essentially, if you zoomed in to any particular region in that, or any particular road in Tasmania, about every car length or so, your genome would differ from the person sitting next to you. And essentially, you could think of that at about every sort of 1,000 base pairs or so, you essentially have a head or a tail coin being tossed at that site. So from that, essentially it then becomes a fairly straightforward statistical test whereby you could add up or sum the number of coins or heads or tails at each of those sites. And that essentially is all that needs to be done by comparing the number of heads and number of tails between people with or without disease, that you can statistically pin down as to whether or not a locus is definitively associated with a disease. Obviously, there are other genomic rearrangements that can occur, and so, for example, you can have major structural anomalies, and this would typically sort of manifest in a severe syndromal disease. Alternatively, you may well have a single site where there's a major genetic change, and this would be a bit analogous to a single major car accident in a position in your genome. But for most common and complex diseases, such as glaucoma, they're probably most analogous just to general congestion on a highway. The other important consideration when mapping or when considering the dissection of a complex disease is investigating the genetic architecture of that disease. And essentially, there are many of the common genetic variants that people harbour generally confer modest or small effect sizes for risk of that disease. And these would generally cluster down in the bottom right corner of this graph, whereby you can see along the x-axis how common that genetic variant is, and along the y-axis how big an effect that particular variant is conferring to the condition. So conversely, the people can sort of harbour a single major genetic variant, and this is a bit analogous to that car crash type variant. But obviously between there are a number of different variants of differing effect sizes which can be mapped. You can then aggregate the effects or sum up the effects of each of these individual variants, and once you reach a certain threshold, disease would typically manifest. Now, obviously, for some conditions, we don't know the actual value that needs to be achieved before a disease will manifest. But for glaucoma in particular, I think we're reaching a point now where we are starting to understand that actual threshold. In terms of the genetic architecture of glaucoma, the Ed Stone's group was actually the first to definitively provide evidence of a genetic cause for glaucoma, and this was where they mapped the GLC1A locus. And then 4 years later, they identified the disease-causing variants in the mycilin gene at this locus. However, it was then not until 2002 that the next major genetic variant was identified, and this was in the optoneurin gene by Mansour Safarazzi's team. But it took another 8 years before the first common genetic variant was definitively associated with glaucoma. And that was by the DECODE group from Iceland. Essentially, from there, every year or so, another gene was identified as being associated with glaucoma. But the next major breakthrough in gene discovery for glaucoma was actually through the release of data from the UK Biobank. And this was really underpinned by the great sort of insight of Sir Pen Kaur and Paul Foster in terms of and they facilitated the release of data on over 100,000 people with both ophthalmic and genetic information. And essentially this has enabled the dissection of genetic or endophenotypes that are associated with glaucoma. So essentially endophenotypes are traits that are associated with or cause glaucoma. And on this, the first sort of major insights were actually harnessed through the interrogation of data related to variation in intraocular pressure. And Anthony Kawaja's team were the first to sort of report on this. And in parallel, we showed that by and large, many of the genetic variants that are associated with variation in intraocular pressure were also influencing the risk of glaucoma. Turning our attention now to the variation in the optic nerve head, and obviously this is the other major endophenotype for glaucoma. And I think before sort of diving into the results from the UK Biobank, I think it was important to reflect on work that David Mackey led through the Tasmanian Eye study. And in that, we mapped essentially a single gene that was definitively associated with variation in the optic nerve head size, and this was replicated in Chris Hammond's twin cohort. But essentially, this took almost 4 years of work to find one single red dot. One summer, a couple of years ago, Jamie Craig and I graded close to 90,000 optic nerve head images, and using that data set from the UK Biobank, we managed to map over 60 loci as being associated with variation in the optic, on the vertical cup to disc ratio. And interestingly, about 2/3 of those loci were novel. We could then, and on that, it was reassuring somewhat to note that the ATOH7 that David identified was also found in this data set. We then managed to mash together all of these traits and case data sets in a multi-trait analysis, and then replicated these findings in the advanced Australian Registry of Advanced Glaucoma, as well as the neighbourhood consortium that was established by Janie Wiggs and Louis Pasquale. What was interesting and terribly exciting is that we identified over 100 loci that were statistically significantly associated with glaucoma, and just under half of these were novel. Importantly, we found no signal for the ATOH7 locus, and to some extent that gives some reassurance that these endophenotype traits can be mashed together and you can statistically tease out as to whether or not that gene variants that are contributing to that trait are also or not contributing to the disease itself. When we look at the effect size of these genetic variants from this mashed up multi-trait analysis, it was somewhat reassuring to see that there was a good correlation to straight disease case control cohorts on their own. We then set about generating and amalgamating these genetic variants into a single risk score, and that comprised close to 3,000 genetic variants. And from that, we could then stratify people's risk of developing glaucoma in the general population. And regardless of the way that you cut the cake, essentially the risk of developing disease based on this aggregated score was very robust. Indeed, when we compared the risk of developing glaucoma based on this polygenetic risk score, it dramatically outperformed that for other tests that have been developed for other common and complex diseases, such as coronary artery disease, atrial fibrillation, type 2 diabetes, inflammatory bowel disease, or breast cancer. And I think that really underscores the fact of the high heritability for glaucoma. In terms of area under the curve, however, from this initial test or initial score, we actually only had a fairly modest AUC. And this has been improved upon over the last couple of years by the addition of data sets from the FinGen group, as well as the Canadian Longitudinal Health Study. But more recently, we've managed to incorporate data from 23andMe, and I'm somewhat pleased to say that this all occurred before they unfortunately had to file for bankruptcy. And this was work that was led by Stuart McGregor and Nick Hahn, but essentially we managed to amalgamate a data set whereby we had an effective case sample size of close to 500,000 people. The Genome-wide Association study of this identified over 600 loci, and in amalgamating all of these genetic effects into a single risk score, we comprised a total of 7 million genetic variants. This new revised risk score has improved this area under the curve somewhat dramatically, and most importantly, this improvement in AUC was actually reflected across people of all our ancestral backgrounds. This increase in AUC was also mirrored by an increase in odds ratio across ancestries as well. And we have replicated this finding across a range of different cohorts. I think what's most remarkable, however, is that when we looked again at Janie Wiggs's Neighbourhood Consortium or Jamie Craig's Registry of Advanced Glaucoma in Australia, we found that if you compared people in the top risk decile, they had almost 100 times greater risk of developing glaucoma compared to the people in the lowest risk decile. So again, I think this is remarkable for almost any common or complex disease to have such a high odds ratio of disease manifestation. The next sort of major sort of point to note, though, is what are the actual clinical implications of this? But I think before diving into that, it's important to reflect about how an individual's genetic risk score has actually changed between the development of each of these polygenetic risk scores. And so here you can see cross-tabulation of the first version of our risk score versus the second. And I must admit, I was somewhat dismayed by the number of individuals whose risk profile had changed. And I think this could be a reflection of either more genetic variants being included in the score. It could be a result of better fine mapping of genetic variants across the genome, i.e. including regions that were not originally included in that original score. Or it could be that the odds ratio for each included genetic variant is actually estimated with less error. Regardless, it's important to note that the majority of shift occurs actually in people with glaucoma. So here you can see or appreciate that between these two sort of different scores that we've described, the greatest shift has occurred in people with glaucoma actually shifting towards the higher risk group. When we look at people in the Blue Mountains Eye study, so this is a population-based study in Australia, we found that only 1 out of 175 people in the lowest risk decile were actually diagnosed with glaucoma. And this compares to almost 20% in people in the highest risk decile. People in that highest risk decile were also diagnosed approximately 10 years earlier than average. And so I think, finally, as a test, we're reaching a point where this could be, or certainly have some utility in terms of population risk profiling. This new polygenetic risk score also outperformed our earlier version in terms of stratifying people's risk for needing to commence or escalate therapy when they're at the early stage of disease. And this was also mirrored by the need for surgical intervention in early disease. The risk profile also is applicable to people with advanced disease, and again, in terms of predicting people's risk of requiring a surgical intervention in advanced disease, has now been replicated across cohorts in Australia and the United Kingdom. So I think we're finally at a point whereby genetic risk profiling for glaucoma does have strong potential clinical utility. However, it's important to note that this is not a diagnostic test, and that it has more utility in actually stratifying people's risk in terms of developing the disease. So with all of that insight, the flip side, or the other sort of major strength that can occur, is actually, can we sort of harness this new genetic information to actually develop new therapies for glaucoma? And in terms of that, I think one of the best examples of the power of genetics for developing potential therapies has been through the genome-wide association studies that have been performed for age-related macular degeneration. And here, essentially, if you had no other understanding about the molecular mechanisms of this disease, and you just designed a monoclonal antibody targeting the closest gene at each of the highest statistically associated risk loci, you would have only had to have designed 9 drugs before you would have developed a blockbuster therapy. Conversely, genetic insight can be used to flag loci or gene targets that would not be sensible to target. And I think a good example of this would be that with genetic hindsight, there's no way that you would have developed a monoclonal antibody targeting the tumour necrosis factor receptor as a treatment for ankylosing spondylitis if you knew that it would lead to demyelination. So in terms of glaucoma, we're now at a point where there are hundreds of loci that could be dissected and used as a new potential therapy for this disease. And I think it would be prudent to use smarter or additional genetic insights before actually going on to develop therapies specifically for glaucoma. And I think on that front, the power of EQTL mapping, so whereby you're mapping the variations, so genomic variations or genetic variants that lead to variation in gene expression, is probably currently the most powerful tool that we have for dissecting out causality of a genetic variant at an individual locus. And the real additional power of this has actually come through the utility of single-cell sequencing. And I think the best analogy in terms of the real power of single-cell sequencing is that sort of good old smoothie analogy. And here you can see a photo of our kids whereby they would often play smoothie roulette, whereby one of them would often hide an unknown ingredient in a smoothie, and you had to taste that and see if you could pick the foul-tasting addition or ingredient. And that essentially is similar to the power of single-cell profiling. So at a single-cell resolution now, it's possible to definitively map variation in gene expression. And so with Joseph Powell and Seyhan Nyaza, we set out to map the genomic regulation of variation in gene expression in our ocular biobank. And with that, we profiled close to 1.5 million circulating blood cells from just over 1,000 individuals. After separating out each of these cell clusters, it was then possible to definitively map the genomic regulations of gene expression. And so initially we just looked at genetic variants that are regulating a nearby gene, and with that we identified over 20,000 cis-acting EQTLs. I think what's remarkable from this is that the vast majority of these seem to be acting only in a single cell type. With this data set, we also had sufficient power to map trans-acting EQTLs, and so these are genetic variants that aren't just acting locally, but also influencing the genetic variation in gene expression in a distal gene. And again, what was somewhat remarkable from this is that the majority of these trans-acting EQTLs appear to be cell and context specific. The additional insight that we could leverage from this unique data set was the ability to map genetic variants that are regulating gene expression across a time course or natural history of a cell. And here, looking at the transition of B naive to B memory cells, we could map genetic variants that were influencing the genomic or gene expression as it varied across that course of the cell. And so the importance of this work or this insight is that it would not be sensible to design a therapeutic target for some of these dynamic EQTLs unless you could target that cell at that specific time and cell state. In terms of disease implications, we then found that approximately 20% of our cis-acting EQTLs actually occurred at disease-specific loci. And using a Mendelian randomisation approach, we could definitively map the causal variants at over 300 genes. And I think this sort of really highlights the potential power of single-cell EQTL mapping across different tissues. The big sort of take-home message from all of this, though, is that I think it's important to map EQTLs across different cells, and especially rare ones, which would generally be missed at tissue-level maps. It's also important to perform this in tissue and disease cells that are specifically involved in the pathology of the disease. And so for glaucoma, that obviously necessitates the need for large disease-specific biorepositories or large induced pluripotent stem cell consortia. And I think that's certainly what I'm excited about in the sort of ongoing work in this area. In terms of this, we're certainly grateful for all of our lab members, as well as the many collaborators and friends that we've worked with over the last 20 years or so. I think it's certainly a great honour to receive this Cogan Award, and there's no doubt that Dr. David Cogan was one of the preeminent clinician scientists of the 20th century. I think despite all of his major contributions to ophthalmology and ophthalmic sciences, I found it somewhat saddening to think that in his obituary in the New York Times in 1993, that the major highlight of his work that was just reflected to the public was his follow-up of people who had survived the atomic bomb. And interestingly, David Nayagam pointed out to me the other day that actually the testing of the dropping of the bomb was actually performed in the Salt Lakes of Utah. So I think with that, with all of the things that are happening across the world at the moment, I think it would be terribly saddening to think that there is the prospect of being able to repeat this work that Dr. Cogan undertook. And to me, that really underscores the importance of ARVO in terms of bringing people from around the world together in a collaborative environment to foster more insights into ophthalmic disease. And I'm certainly very excited about the future, both of ARVO and of work in ophthalmology. So thank you.